Okay, so thank you for the invitation and the kind introduction. And now, I have the opportunity now to show what we are doing, what we have been doing in the last few years at the Magnetic Resonance Center of the University of Florence. We are traditionally a group dealing with NMR of metalloproteins, but over the last 15 years we started also looking at metabolomics, which is a completely different approach in the use of NMR. And now we are trying to combine the two aspects to learn something about the chemistry of the systems that are interesting for us. So metabolomics is a tool that provides information on the downstream of the effects of the various omics, and at variance with the other omics looks at a very heterogeneous class of molecules which have in common only the size, because the definition that you find on the human metabolome database, for example, means that the metabolome of an organism of a given sample is made by all the molecules that have a molecular weight below a certain threshold, usually 2 kilodalton. So these are small molecules that have different origins and are very different from the chemical point of view of their composition. And we can work with different types of samples. We can use, if you stay on the human or animal part, we can use with biofluids that can provide an overall fingerprint of the status of a given individual and can provide information about effects of therapeutical treatments and so on. But clearly is a very overall and general effect that is difficult to interpret in terms of biochemical point of view. Something intermediate could be when we look at metabolomics of tissues or specific biofluids like cerebrospinal fluids, because they're mapping what is going on in a given organ or a system. Apparatus. Then there is metabolomics of cells. Metabolomics of cells, if you look at cell culture, you can monitor which are the changes following a given treatment inside cells, and also the exchange of molecules with the growth media. And my talk will be focused on this aspect. And this is just the workflow that we use when we want to look at the effect of given drugs in a given cell culture or cell line. So we measure the C50 just to have information about the cytotoxicity of the molecule. Then we treat cell cultures with different drugs for different times, but using equal toxicity conditions, so using the IC50 measured at 72 hours for each of these compounds. And then we collect the cell lysates and the growth media, and we analyze them separately, and then we try to learn something about the biochemistry of the system. Just to give you an idea of the spectra, these are simple proton monodimensional spectra, because clearly if you want to screen several conditions, several drugs, we need something that can provide information quickly and without too many difficulties in their analysis. So if you look at cell lysates, usually we can use different pulse sequences, and we get about 30 metabolites that can be safely assigned and quantified. A similar number if we look at the growth media, but only some of these metabolites are in common. So usually we end up with about 50 different molecules for a given sample. Clearly, each cell line has its own pattern, which usually is not determined by different natures in the metabolites, but different relative intensity, which means different relative concentration in the different cell lines. And when we treat samples with drugs, what we see are changing in the relative intensities, because some pathways are upregulated and others are downregulated by the treatment. So NMR, as has been discussed before by the other two speakers, is relatively low sensitivity technique, so the numbers are not very great. But if you look at the pattern of the biochemical pathways that we can have inside the cell, these about 50 metabolites cover a large part of them, although none of them is really selective for a specific pattern because metabolites are involved in many different patterns. But still, if we want to have an overview of the different effects of the metals, of the metallodrugs, we have an idea. So, for example, these are the three most commonly used in clinics: platinum drugs, cisplatin, oxaliplatin, and carboplatin. And if you compare which are the effects at the level of the cell lysate and growth media, you see that there are very common patterns. So here you have 24 hours, 84 hours, 48 hours, and you see that you need some times before observing the largest effect, but for example, at this level, the three compounds behave in the same way. So we can see that the dots, the asterisk indicate, of course, those that are statistically significant variations. If you look at the exometabolome, so at the growth media, you can distinguish between those metabolites that are released in the media, so they are not present at time zero, but are released by the cell during this growth, or those that are uptaken from the media, and so they are reduced in intensity over time. And so you can derive some hypotheses about which are the pathways that are mainly affected by the treatment. So what we learn looking at these platinum-2 drugs is that, in general, they have a different degree of effectiveness, but they behave more or less always in the same way. For us, more important was to look at a series of gold drugs that are developed by the Department of Chemistry. They include drugs which are FDA approved and new candidate drugs where people are played with different ligands and so on. And they include gold 1 in yellow and gold 3 derivatives. They have different IC50 values, so apparently they don't have many things in common. But if you look at the metabolomics pattern, you can identify differences and families of drugs that behave in the same way, independently of the IC50 value. Again, we are always working with the same under equal toxicity conditions, so we use the IC50 value measured at 72 hours for each of them. And so what we see is that, for example, auranofin, which is a drug that is activated by losing this tire sugar and maintains the phosphate ligand, has very large effects already at 24 hours, and in particular, one of the main effects is the increase, a large increase in glutathione concentration, which has no counterpart, for example, in the group of these gold carbene compounds that instead have a reduction in glutathione. For aurotiomalate, which is this other drug, gold 1 drug, we have an increase in glutathione, which is slower, appears later in time of the treatment, and is never so high like for auranofin. So, working on this type of analysis, we could group the 9 drugs that I was showing before in different families. And so we see that all the auranofin and its derivatives are more or less inducing the same type of effect: large increase in glutathione, early and large, and then this reduction in the release of pyruvate and slight increase in lactate, while in the other class we have a large increase in lactate and decrease in glutathione, and here we have effects on the TCA cycle. So, at variance with the case of platinum-2 drugs, in the case of gold 1, the nature of the ligands is modulating the effects. This is not unexpected in the sense that in the case of platinum, the target is always DNA. In this case, the targets are sometimes not yet identified cytosolic proteins. And so the nature of the ligands can modulate the affinity for the different type of proteins. Identification of proteins is not an obvious task, so we are trying to combine different omics and also genome scale modeling, trying to understand which could be the target protein. And proteomics and metalloproteomics seems to provide the most interesting aspect. But staying on the aspect of NMR, okay, these are the effects that are induced in certain ovarian cancer cell lines. We are trying to reproduce, and we are observing the same effects on a different ovarian cancer cell line. But when we have these drugs, they are not selective. Metallodrugs can bind to proteins that contain specific solvent-exposed ligands. For example, in the case of GOLD1, the most interesting proteins as potential targets are those that present cysteines or selenocysteines that are accessible to the gold. So usually metallodrugs don't have a single protein target, but have many different protein targets. And as I was showing, the nature of the ligands on the gold drugs can modulate the affinity for the different proteins. We want to have something which is more selective against cancer cells. So we wanted to use some sort of nano-delivery of these drugs, trying to increase the selectivity towards cancer cells. And the system that we selected in this sense is ferritin. Ferritin is present in the cytoplasm of all animals as an heteropolymeric cage, nanocage protein of 12 nanometer of outside diameter. And this 24-mer cage is generally composed by two different types of subunits, which share the same type of fold but slightly different sequences. One is, which is called H, has enzymatic activity, so is able to oxidize iron very quickly and then induce biomineralization inside the cage of iron oxides, of ferric oxides. In the case of the L-type ferritin, which is slightly shorted, she misses the catalytic site, and so is able to facilitate biomineralization by providing some nucleation sites on the inner cage surface, but has no catalytic activity. So if you build up a recombinant protein which is fully aged, this is very active in iron oxidation. The other, if you have an homopolymeric L cage, is very slow in biomineralization. In nature, depending on the tissue and the health status of an individual, you have heteropolymers with different ratios. But we were interested in using human H ferritin, so a pure recombinant H protein to be administered once loaded with our gold drugs. So we have a very large external subunit. So the idea was not to use the inner cage that requires disassembly for incorporation of the drugs and usually induce loss of large amounts of protein, but just to exploit the surface, like people usually do with monoclonal antibodies, okay? So we did the reaction with the different GOLD1 compounds that I was showing before, incubation for 3 hours, 37 degrees, dialysis to remove excess of gold, and then characterization of the product by mass spectrometry, electron spray mass spectrometry, and ICP to determine the binding of the gold to the single subunits and in the total metal loaded on the cage, and then further analysis using also nutritional studies. So first of all, if we use MS, we look at the single subunit, because the treatment that we have of this cage with electrospray induces disassembly. Okay, but we can monitor, combined also with trypsinization, we can monitor the type of species that are bound and also the binding site. To validate our proposal of binding sites was we use also mutations. Because in each single subunit of the H protein, we have 3 cysteines, which are good targets for gold 1, is Arden's soft theory. 2 are very well solvent exposed, and 1 is buried inside channels that are connected the inner cage to the outer cage. And what we saw that is that if we stay with these 2 compounds, we have only binding to the 2 solvent exposed cysteines, while in the other case, if we use a quite large excess of these compounds, we can have also partial metallation of the cysteine that is inside. Okay, so we had an idea, but the problem of this approach is that you disassemble the cage. So you may lose metal ions that, or metal compounds that are entered the cage, if possible, or also metal ions that are bound cooperatively to pairs of subunits. And so we combine this with ICP on the intact cage to validate whether the amount of metallation that we determined by mass spectrometry is compatible with the ICP analysis. And this was the case for these 3 compounds. So why human age ferritin could be a good carrier for targeting cancer cells? Because, as been reported by several years by many authors, it can bind to transferrin receptor 1 and can be internalized following the classical endosomal lysosomal pathway. And if we look at cancer cells, and in particular to our cancer cell line of reference, you see that the levels of TFR1 expression are much higher in cancer cells than in healthy cells. And indeed, we are able to internalize a large amount of ferritin in the case of our cancer cell line with respect to the healthy cell lines. So this could be a targeting, but then what about the metabolomics? We have spent many efforts in identifying the patterns that are altered by the single drug compounds or drugs or drug candidates. What happens if I treat cells not with a free compound, but with a compound bound on the surface of ferritin? And these are the results for auranofin. You see, you have in green the patterns that we have for the conjugate, in yellow the free drug, and in white you have the apoferritin, so the ferritin without any metal ion bound, so that to exclude large effect that could come from the protein cage. And you see that the patterns are exactly the same. These are the meaningful changes are those highlighted here, and they are the same at the level of the lysates and the growth media. So with auranofin, it works. So this could be a good carrier. What happens if I try with aurothiomalate? Aurothiomalate is this molecule that in solution has very low solubility because it's sort of polymeric structures, helical polymeric structures, but it can bind in very well defined ratios with the protein. And again, you see, in blue you have the conjugate, in red you have the free aurothiomalate. The pattern induced at the metabolomic levels are the same. So, what does it mean? It means that not only we can target cancer cells by ferritin and with higher affinity for the with respect to the healthy cells, but this carrier does not alter the metabolomic changes that are induced by the compounds. We wanted something more at the level of mechanism of action. So we treat cells with these bioconjugates. at which level the drug is released to its intracellular targets, when the protein is disassembled. I mean, the protein bioconjugate is disassembled. So our idea, being mainly NMR spectroscopists, was to look at NMR spectra. Because clearly, if we put a fluorophore on the protein, we see where the protein is going, but we don't have information about gold. We can measure the total amount of gold internalized by the cells, but we cannot measure how much of this gold is bound and what is the kinetic of the release. So, NMR. NMR, we were been working a lot about 10 to 15 years ago on a different type of animal ferritin that shares a very large homology with the human age, and we have been able to acquire significant NMR spectra, but there are many drawbacks. First of all, this is the size of the protein in terms of size in nanometers and molecular weight. So it's really huge. It's huge also for the artificial intelligence approaches that were shown before. And so this affects the tumbling time, therefore the T2 relaxation time, which means broadening of the lines, induce a very large broadening, and so we cannot build up the scalar couplings that are needed for the sequential assignments. So in order to assign a large share of, but not complete assignment of this protein, we had to move to solid state NMR and then transfer the assignment to the solution. And the most powerful approach that we could obtain in solution was to work with NOE, because low tumbling proteins have long T1, if they're very slow tumbling, like ferritin, and so this induces very efficient NOEs, but not proton-proton NOEs, nuclear effects, because the signals of proton were too large, but carbon-13. So carbon-13 allowed us to look at the spin systems of the side chains. So this type of spectrum was very useful for us when we wanted to study the mechanism of action of ferritin in its natural context. So how this is internalized iron and the formation of the ferric products using paramagnetic NMR. You have heard this morning paramagnetic NMR induces signal broadening, and in this case we could observe signals that were broadened beyond detection due to this formation of the ferric products, and using the assignment made at the solid state and transferred to the carbon-13 nosy, we could monitor the pathway of this iron 3 species. But if we want to screen our drugs, which are many drugs, under different conditions, especially if we want to look at the cell lysates, this is unfeasible. Also because everything in solution was working using per-deuterated protein, so 90%, at least 90% deuterated ferritin, which means very, very expensive samples. So we look at our model structures or solid structure. So this is the structure of the apoferritin. I'm just a close-up of one region of a single subunit which contains the two cysteines indicated in orange. This was the structure that we got with one of the gold monocarbines by cryoEM, 1.51 structure, very well resolution, where we see a cluster of gold ions. And these are the model structure obtained for auranofin and orothioamalates. So, if you look, these are the cysteines. In yellow, you have the gold ions. Here, you have in gray the other ligands. But what you see that there is a tryptophan very close to these cysteines. And this is the only tryptophan in the sequence of the subunit. So, we decided to use fluorine-19 NMR by producing a ferritin cage that is labeled in with fluorine on the tryptophan. And this is very easy, just using a 5-fluoroindole and the production of the proteins like for the wild type, so that we have no problems of cost or troubles in the expression. And then, although the protein is very huge and large, what we have is that if we look at the apoprotein, we have a single signal that represents the status of the tryptophan in this protein. We have a single signal because the 24-mer cage is highly symmetric. So I have the signal from all the cages, from all the subunits of the cage that is exactly equal by symmetry. If I use, for example, auranofin that is, can be only partially metallate the apoprotein, I see two separate signals. So the exchange is slow on the NMR time scale for the apoprotein and for the protein bound to the auranofin. Or if I look at the monocarbene, for example, this is the apo, this is an intermediate species that I obtained before I have the formation of the full gold cluster, and this is the final adduct. So I have different signals that represent the metallation status of my protein. And so the idea now is that we can monitor in the growth media, we can see the uptake of the free ferritin and compare it with the ferritin metallated and see whether the presence of the metal ion somehow interferes with the interaction with transferrin receptor 1, which needs to internalize. But with the new instruments that we have now, with fluorine 19 dedicated probe, we could go down to 5 micromolar concentration of ferritin in the samples with observed a spectra of the quality of the fluorine in 1 hour. So now I think we are ready to look at what is happening to the ferritin bioconjugates inside the cells, but just looking at these fluorine 19 signals. So that's my conclusion. I think that NMR-based metabolomics provides a well-defined fingerprint of the cellular effects of metal compounds, that in the case of human H-ferritin, the solvent-exposed system residues provide a very high affinity binding site for gold-1 compounds, but also for other soft metal ions. These bioconjugates can be internalized in cancer cells via the TFR1-mediated process, and they maintain their cellular effects as shown by metabolomics. Clearly, the characterization of these bioconjugates in terms of purity, behavior with respect to competitors and so on is not obvious and requires integrated structural biology approaches. In our case, mass spectrometry, ICP, molecular dynamics, cryoEM, very powerful because we have a good resolution around the heavy metal ions, and also now fluorine 19 NMR. These are the acknowledgements to the people. So these are people at our Magnetic Resonance Center working with me, Veronica, Lucrezia, and Stefano. These are other colleagues which are mainly on the computational part, so molecular dynamics and help in solving the cranial structures. And these are the colleagues at the Department of Chemistry who are producing the metal compounds and doing the mass spectrometry analysis. And this is funding from the Italian Association for Cancer Research. And thank you for your attention. And finally, sorry, Nicola Brescia in Bilbao for helping in the cryoEM resolution. Okay, thank you for your attention.
We have time just for one question, if there is one question. Otherwise, we move to lunch so we can come back at the right time for the afternoon session.
There is a question over there.
Just one. Yeah, go.
Very quick one. It was a really nice talk. So when you take your ferritin, if you label it with the methyl groups and in deuterated background, how does the spectrum look? Is that something you could potentially use?
Deuterated background, not for proton NMR.
So even the methyltrosine.
I see, yes, even the methyltrosine, even the creneptrosine provides a number of signal which is really low with respect to the number of expected signals for a protein of with subunits of this size. So the way that we use was to move to solid state NMR, where the T2 coming from the tumbling is not a problem. Then assigning them, and we were not able to assign everything because this overlap is not that obvious, and then transfer this assignment to the per-deuterated sample in solution, but just using the carbon-13 nosy, which is not obvious, but we were lucky because the pattern are preserved between solution and solid state, which is not that obvious in terms of chemical shifts. Okay.
Thank all the speakers.